All right, so as we've noted, this is sensation and perception, right? Which is going to be a fun class for you. Okay. I do want to give you just a bit of an idea. I always try to start with a little bit of context, right? Some of you who've been in my classes before, you kind of know where this is going. For those of you that haven't, though, I think it's kind of a nice orientation, right? So you can kind of know what's going on, how I lecture. My style might be different than other folks, right? And I think it's important for you to know what's happening so you're not confused any more than you're already going to be confused, right? It's like this baseline confusion you're going to have. I don't really want you too much above that, right? And you need to stay right there in that. So what I like to say is this is kind of like a carousel. Right? We're going to revisit a lot of topics in this class. Probably one of the number one phrases you're going to hear me say is, do you remember that time I told you? And then I'm going to say something that I've already told you, right? And then we're going to go right back and we're going to try to connect those thoughts and those ideas together, okay? So if you happen to fall off the carousel, that's fine, right? We're going to come back around and we'll pick you up again, right? If you get so far off that you just think you can't find your way back, that's when you can ask a question, right? That's when you can stop by office hours. That's when you can send an email. That's when you can check your textbook and read things, right? You've got a ton of resources here. We're really trying to provide you with all of those resources so you can be successful, okay? I also like to think about this in layers, right? Sort of a hierarchy of understanding. So there are going to be things that we're going to throw out there sometimes that aren't going to make a lot of sense to some of you. They're going to make more sense to others, and that's going to fluctuate throughout this semester, right? Those of you that are musicians, I ask that for a very real reason. When we talk in a couple weeks about sound perception and the auditory system, some of the things that we talk about there are going to make much more sense to those of you who have played an instrument, right? Who understand what a B flat or a C sharp is, or the difference between a clarinet and an oboe, right? And so we're going to talk about those things on some level. For those of you that are like, don't know what an instrument is, you're going to struggle a bit more with that. But maybe you're going to have something else later that's going to come in and it's going to help you, right? So keep that in mind. Just hang in there. It's going to kind of come and go. The other thing to keep in mind is I like to let people know I'm a reductionist, right? It means I kind of go for that bottom end layer, right? So in this class, we're going to talk a lot about, we're even going to talk about physics, right? We've got a lecture on the physics of vision. Right, I'm going to talk to you about light waves and particles of light. We're going to talk about the very physical stimulus, right, that's going to interact with your nervous system so that you can have this interesting sort of experience and perception about the world out there. And so I think that's something to keep in mind, right? And so when we do that, just hang in there. It's going to be fun. It's going to give you hopefully some deeper understanding about how you interact with the world, right? I think that's going to be helpful. How many of you are psych majors? Anybody who's not a psych major? Just one? Okay. Andrew, what's your?
Health science.
Health science, okay. Yeah, I get a lot of communications disorders folks too who take this class, which is helpful. And then now that they've got the aviation program, apparently they're all going to funnel through this class too. Yeah, extra work for me. But not for 2 years, I think, right? We're not going to, because they won't take upper division classes for a while. All right, let's think about general principles. So why should we even study the senses, right? That's kind of a good question. And one of the reasons you should study the senses is because it's like a required class for you to graduate, right? You got to take one if you're a psych major out of that category, right? And you thought this was going to be the best option. Well, little did you know it was not, but you're here anyway, so let's just deal with it. Perception, for most folks, we'll talk about some interesting cases when it's maybe not the case, is very effortless, right? And it's immediate. How many times have you looked at a piece of fruit and you've had to really work to figure out what color it is? You don't, right? You look at it and you go, oh, that's a red apple. That's a yellow banana, right? It's an orange. That's the easy one. So perception is very effortless. It's very easy, right? We usually don't make mistakes, okay? Most of the time it agrees with other people, right? Our sensory systems are built, on some level I'm going to tell you they're identical, and then sometime later I'm going to tell you there are some minor variations, right? And we'll talk about those variations. But by and large, our perceptual experiences are identical because our sensory systems are identical. And we're going to be receiving the same physical stimulus, right? If your eye, and the back of your eye, we'll talk a lot about that, if those rods and cones, you guys are probably familiar with those photoreceptors, right, on some level, if they work the same as everyone else's, then the same frequency of light that comes in, that same wavelength, should be perceived as the same color by everybody, right? It just makes sense. If it's the same, receiving apparatus and it's the same stimulus that gets to you, then everything should be the same, right? Okay. In fact, though, perception is really complex, and there is a bunch of your brain that's devoted to just processing sensory signals, right? Probably over half of the outer part of your brain, called the cortex, is devoted to sensory processes, right? So over half of your brain, in some sense, is devoted to just receiving these signals from the outside world and processing what those signals mean, okay? So a lot of brain function is devoted to that, right? Which is really kind of interesting. Additionally, sometimes there are errors in judgment, right? Particularly when we're in critical situations, okay? Some of the very early sensation and perception work was focused actually on aviation safety, right? So when folks started flying more for either recreational, military, or commercial reasons, those pilots have a different experience, right? What's the fastest you can move without the assistance of a machine? Like, how fast can a person run, right? That's my question. 20. I mean, I guess it depends on what's chasing you, right? Let's say you can run 20 miles an hour, right? I mean, I don't know. You can't do that for very long, necessarily, right? Can you imagine the marathon times if you could? Yeah, it'd be ridiculous, right? But let's say even 10 miles an hour, 20 miles, even if you go up to 30 miles an hour, right, for a brief period of time. How fast does your car go? How many of you know it goes less than 30 miles an hour, other than when it's parked, right? Most of you don't, right? Our systems aren't built to travel 70, 80, 500 miles an hour, right? But yet we have to make those judgments traveling at that speed, right? And so you can see why that becomes a real problem. There's actually, there was a lot of work done in aviation safety. How far apart, what's the minimum distance needed between two planes in order for the pilots to miss each other, right? If you get so close, there's a certain point where you're not going to have time to respond and make a corrective adjustment, right? And the same thing in cars, right? Once you get so close to another car, impact is inevitable, right? If they're coming at you head on, because you don't have the time, neither does the system you're trying to drive have the time to respond to that, right? So these are very important things to consider. Okay. We should think about some basic definitions. We talk about sensation and perception together often, right? Most folks say sensation and perception. It's the title of the course. In fact, they're two separate but connected concepts. Sensation is simply that stimulus-receptor interaction, and we'll talk a bit more about what we mean by that stimulus-receptor interaction. We've given you a basic idea. We talked about light, and then we talked about rods and cones, right? That's a receptor. Okay, just that interaction, that's sensation. Now, what you make of that is perception, right? When you go beyond that, when you add some interpretation. The problem is it's a little difficult to draw a line between the two, right? Usually you can tell what is clearly perception when you're thinking about, for example, that's a red apple. That's clearly perception, right? You're interpreting those signals, okay? The light wave or particle hitting the photoreceptor, that's clearly just sensation, but very quickly you get into sort of a fuzzy spot in between the two, right? And you have to try to work your way through that. And there are folks who debate about that constantly, right? Like, where is that line? Does the line exist? Should we think about it as a line? I think for our purposes, let's think about the things on the extreme so we can kind of know what the difference is, and we'll let the stuff in the middle fall where it does. All right. So when we talk about senses, we generally talk about what we call a modality. Okay. Right. There are sort of two broad groups of senses. We have what are called the general senses and the special senses. Under general senses, we think of things that are somatic or visceral. These usually relate, you can kind of think about the body. Okay. Visceral senses we're not typically aware of. Right? These are things that are going on internally. We won't spend a lot of time thinking about that in this course because they're not really things that enter our perceptual world. Right? How many of you currently know the concentration of oxygen that's dissolved in your blood? Nobody does, right? Your body knows on some level if that's at the appropriate level because you have a sensory apparatus for that, right? So there's definitely sensation going on, right? There are receptors that monitor your blood. They monitor how much oxygen is in there. They monitor the pH. They monitor all kinds of stuff, right? But they're not things that you're generally aware of. Okay, so visceral senses we're not going to think a lot about. Somatic senses, these are things like pain, hot and cold, touch, pressure, vibration, tickle, itch, and then proprioception and kinesthesis, right? So this is what's your body doing when it's at rest, and then what's your body doing while it's moving, right? How many of you are aware of where your feet are? But how many of you are actually looking at your feet? Probably not many of you, right? You know where your body is, you know the position of your body. That's proprioception, right? When your body starts moving, then we've got some, kinesthetic element there, and you're aware of that as well, right? Most of you probably don't have to look at your legs while you're walking, okay, right? That's helpful so you don't run into trees. It's kind of a nice feature. So there you go. So there are your general senses. When we think of special senses, we look at vision, we've got hearing, equilibrium. We'll talk a good bit about the vestibular system, right? So what we think about there. So you know balance, you know which way's down. That's always important, right? Smell and taste. So those are sort of your special senses. We will spend most of our time in this course focusing on somatic and special senses. In particular, we'll spend a lot of time talking about vision. As humans, we are highly visual species, right? We rely on vision much more than we rely on a lot of our other senses, and we rely on vision more than some other species rely on vision, right? If you think about bats, for example, they rely on vision some, most certainly, but auditory stimuli are much more important to them than they are to us. Rats, for example, olfactory stimulus is important. Most of us are not going to be able to smell our way around, right? How many of you have to go to another building after this? Yeah, I challenge you to close your eyes and just try to smell your way over there. It's not going to work, right? You're not going to get very far. You need to kind of look where you're going because that's what we rely upon. Now, there are obviously situations where you can use other senses to get around. Here's sort of a breakdown of the senses, right? If we wanted to classify these, here's the sense, what stimulus is responsible for that. We'll talk about each of those in detail as we cover that sensory system throughout the semester. We talk about the kind of receptor. Notice there are different sort of receptors. Each one is going to be perfectly matched to the stimulus that's coming in, right? Vision, for example, you're trying to measure these hearing. You're trying to measure airwaves. So you have these receptors in your ear that get kind of shaken, right, when those airwaves come in, right, when those vibrations occur. It wouldn't really work if you had things in your ear that were sensitive to light energy, right? If you had light coming in your ear, that doesn't seem to be a very effective way to get around the world, right? Okay. So we want to keep in mind that the receptors match to the stimulus. And typically we have a place in that outer covering of your brain, in that cortex, where it's going to be a target for each of these sensory systems, right? That information is going to come in, We'll talk about this. You do a little bit of low-level processing, and then we're going to keep sending that up to higher and higher places, right? Because we have to take the information you're receiving. Doesn't seem very effective this way, but it breaks down. Your nervous system breaks down the information coming in and then reassembles that information. So it's kind of an interesting process that we're going to go through multiple times. Not entirely sure what happened with the picture here, but we'll get that corrected. This is just an example of different sensory receptors, right, based on structure. So there are ways you can classify these receptors, and one way you can do it is based on the structure of the receptor. This has a free nerve ending. Notice there's nothing down here on the end of that sensory neuron. That's really great for something like pain perception. You want that information to sort of directly and quickly get to its destination, right? You don't want to wait until the saw is halfway through your hand before you realize you're in pain, right? If you can get that signal there faster, you're not going to cut through as much meat, right? There are other sensory receptors that may be encapsulating that. Makes some sense, okay? This would be something for vibration or pressure, right? Can you imagine if your like pressure sensors were very, very sensitive, and every time like a fly landed on your hand, it felt like somebody was squishing your hand with a brick? Doesn't make any sense, right? You actually want some sort of resistance there. And then finally, we have specialized receptors. This is an example of a rod that's going to be found in your eye. We'll talk about that in detail. Sometimes we need very specialized structures to receive that sort of stimulus, right? So in this case, we're going to have light waves coming in. We have to have that specialized structure to catch those, okay? We can also classify receptors based on their location. There are ones that are sort of on the outside, ones that are on the inside, and then we have a special group that's in the muscles and the joints. And then finally, we can classify them based on the receptor or the type of stimulus. We have mechanoreceptors. These are things that you typically find in your skin, right? They're sensitive to mechanical pressure, right? So again, some sort of pressure, vibration, texture. Thermos, hot, cold. Nociceptors, those are pain. Photoreceptors, those are sensitive to great pictures that you take. It's light waves. Chemoreceptors, they are sensitive to chemicals. What kind of chemicals would we think about? This is going to be for taste and smell. And then osmoreceptors, these aren't things that you're necessarily going to be aware of, but they measure things like blood pressure. Okay, they would measure osmotic pressure, right, in your body. So that's not going to be something you're necessarily aware of, but it's there. All right, let's talk about this guy. His name's Johan Mueller. Johan Mueller's a pretty cool guy. He looks like a real happy dude. You go out and meet him. He seems like he's, you know, jolly old Johan. So why is Johan Mueller so important to this discussion? And it's this idea he had called the doctrine of specific nerve energies. Basically, the idea behind this is every signal, once it gets in your nervous system, once it gets past that sensory receptor, whatever that is, everything that comes in is just an electrical signal, right? It's an electrical signal that we call an action potential. Okay. Some of you may be aware of this. Hopefully in an intro to psych class, someone talked to you about action potentials, right, briefly. I'm not going to ask you to know a whole lot about action potentials, but we need to think about it just a little bit, right? So, if everything is just an action potential, if once you get into the nervous system, everything's just this electrical signal, how do we know if something is light or if it's sound or if it's touch, right? And the important thing here is the destination, right? Is this going to visual cortex? Is it going to auditory cortex? Where is it coming from and where is it going? That's how we know what it is. Because the signal's all the same, right? It's all just an action potential. some change in an electrical signal. They all look identical, right? They're all the same. You wouldn't have any way to know if I showed you this and I said, hey, what caused that? You have no idea. You have no idea if it was a light wave, if it was a sound wave, if it was, biting into a hot dog. Could be a million things. Okay, so it's the destination, right? So start and finish is what's important. For those of you that have not seen a drawing of a neuron in a while, here's kind of a brief reminder of what that is, right? When we think about these action potentials, they go down this axon. This area is called the dendrites. That's the part that sort of receives signals from other cells. And then we have the terminals down here that help convey a signal on to another neuron and send that signal going through. You're not going to have to know a whole lot about how this process works, but some basic idea might be important for this, right? So if you kind of review that, especially looking over those particular parts, that might be something of benefit to you. All right, let's skip that slide. But we should think about something called thalamus, and this says cortex. Okay. These are sort of the two major divisions in your brain that are going to handle incoming sensory signals, right? And we'll talk about specific subdivisions of each of these regions as we go, but for right now, let's think about them kind of broadly, right? When you have a sensory signal, whether that's coming from the eye, the ear, the body, the first destination is going to be the thalamus, okay? It's this guy right here. It's a sensory processing location sort of in the middle of the brain. It handles very sort of basic rudimentary information, sort of low-level information about the sensory stimulus that's coming in. Once it receives that information and it processes that, it sends that information up to cortex somewhere. Again, that's going to be specific to the sensory modality. Okay. Now this is a very general story. I'm going to give you this specific story again and again and again and again and maybe one more time. I was trying to think about how many sensory systems we're going to discuss. Each time we discuss a sensory system, it's going to be the same basic concept, right? Sensory receptor, information goes to thalamus, that information goes to cortex. Okay? Interestingly, information from cortex goes back down to thalamus, can kind of change the way that that structure receives information and processes information. We'll talk a lot in this class about something called gain control. Okay. Don't expect you to know a lot about that today, but that is something that's going to be kind of important. How we tune our responses, right? How these brain regions that are receiving sensory information, how do they adjust, right? A good example of this we'll talk about in detail later is something called color constancy. How many of you are aware that grass is green? Right, and that's pretty basic, right? Great job. Thanks for the enthusiasm. It's green almost all the time, right? Whether it's kind of overcast like today, or the sun's bright, or it's getting close to nighttime, the grass is still green, right? And you still know it's green, you still see it as green. How do you still see it as green? We'll learn later that the light source hitting an object has a major impact on how that is perceived by you, but at the same time you also know grass is green, right? So these higher order brain regions are sending that information back down and they're saying, hey, grass is always green, let's adjust our scale so we know grass is green, and then we'll work around that, right? Don't worry too much about this. All right. We should think about how we study the senses. There are a number of ways that we do that. Okay. And we'll talk about sort of each of these images or each of these approaches as we go. Probably one of the more important approaches that we'll think about are psychophysics. Sounds very exciting. It can be quite boring, but it's still kind of an important approach. All right. This guy, Pierre Florenz, he had a technique called ablation. The basic idea with ablation is he's going to remove part of your brain He did this in animals. Remove part of the brain of an animal and then see what that animal can or cannot do, right? If you remove part of the brain and the animal can't walk, that must have been the part of the brain that controlled walking. There you go. We figured something out, right? The problem with this is this guy wasn't very precise, right? He was going on early. He just kind of took a spoon, scooped out some stuff, and was like, oh, let's see what happens. Again, this was the first guy to do it, right? Somebody goes at a spoon with your brain, goes with a spoon to your brain today. Doesn't sound so great, right? But we have much more advanced techniques now. The reverse of this is an idea where you might have humans who have accidents, some sort of neurodegenerative disease. Think about like Alzheimer's or a stroke that causes brain damage. So this is the reverse, right? Rather than actually causing the damage to the individual, it doesn't seem very nice. We wait until the individual has some damage, and then we can figure out, later what that brain region was responsible for. Some of you have heard of Paul Broca, right? Or Broca's area, Broca's aphasia, right? It's a pretty common thing folks talk about. This was his idea. He learned pretty much everything from this individual. They called him Tam. That wasn't probably his actual name. This individual had some brain damage, and the only word he could say after that was Tam, T-A-M. So much like Hodor, if that's the only word you can say, that becomes your name. So you sort of get stuck with that. So he got stuck with the name Tam. Once Tam died, they cut his head open. They're like, hey, this guy couldn't talk in the way that other people do. Let's see what happened. And there was part of his brain was missing. Broca was like, that's called Broca's area, because I was the guy who was, checking this out. And so that's how we get the idea. So it's kind of the reverse. A lot of the work was also sort of to define this much more was done in Japan, actually, in the early 1900s. There was an individual there. His job was to, sort of an interesting job, during the Russo-Japanese War, when Japanese soldiers were injured, they had bullet wounds to the head, they would get some sort of pension from that, right? And the amount of money that the military would pay them was based on the extent of the damage that they sustained in combat, right? Makes some sense, right? So this guy, anyway, he developed a device to measure how much of the visual field was lost by each individual person who was shot in the head, and based on how much of the visual field was gone, how much blindness they experienced, and they would pay out more money. What was interesting is he was able to map out how much of the visual field was gone and where the damage was in the brain, and so he was able to make a direct connection between the location of brain damage and the location of the visual field deficit, right, in these individuals. This was a huge breakthrough because this told people that there is a map of the world directly represented on your onto your brain. So this again was a very important breakthrough. And so he did this for a while. We've since refined our understanding of these what we call topographical maps. Right? Most sensory systems have some sort of mapping feature. Right? So for something that's spatial, like your visual system, having this topographical map makes a lot of sense. For the auditory system, it doesn't quite make as much sense to have a topographical map. In that case, things are mapped out based on frequency, so it's a tonotopic map. We'll talk about other mapping features as we go. One of the other interesting things that came out of this was a concept called cortical magnification. The idea here is part of your world is more important than other parts, right? And in the visual system, that very center part of your vision, right, is the most important. And we'll talk about why later. That few degrees of space gets a huge amount of space in your brain, right? Where what's at the periphery, even though it takes up much more real-world space, gets much less real estate in your brain because it's not as important, right? And so we'll talk about this concept of cortical magnification as we go. Just want to sort of introduce that to you now. Some of the studies we'll talk about involve what's called microelectrode recordings. Basically, there were some folks who took a thin wire, they stuck that into a brain cell, and they could measure those action potentials, right? And then what they could do is they could present a stimulus of some sort and see if that cell got excited or not, right? Okay. If it got excited, then we could say, hey, that brain cell really likes blue light or green light or bars of light that are at a certain angle, right? One of the problems with this is you're only looking at one out of like a trillion cells, right? It seems kind of oversimplifying, right? How many of you have like a broad collection of music, right? Anybody? Yeah, right. And let's say you have 1,000 albums. I'm just going to make up a number, right? And let's say one of those is polka, right? 1 out of 1,000, and the rest are like some other genre, non-polka, right? Should we look at that one album? Should we pull out that one album and assume that you are like a big fan of polka? That's probably something your grandma got you for Christmas, right? And you just kept it because you felt sorry. So we're not going to judge you on that, right? So maybe that's an oversimplification, just looking at one out of like a trillion cells. So then some folks said, well, hey, why don't we just like record from a bunch at once? And they made some technological advances, and they were able to record from maybe even 1,000 of these guys at one time, right? It was really impressive. You can skip these, not so important. Another method is imaging, right? Most of you are familiar with CT or MRI exams, right? fMRI. These are just different ways to image what's going on in the brain. There are a few other whole brain methods that you would do, like an EEG. If you've had a head injury and you've had to go to the hospital, they'll probably give you an EEG. If you've done a sleep study, same thing. It's just a way to measure that electrical activity of what's going on in the outer part of your brain. You can also do this with big magnets, right? For those of you that are really into physics, you know electrical fields create magnetic fields and vice versa, right? So instead of measuring like electrical signals, you could potentially measure magnetic fields as well, and the way those change, you get some idea about that as well, right? Okay, so that's exciting. But let's spend a little time talking about psychophysics, right? This is really a way, and there are a few approaches, but this is really a way to measure what's going on, right? And so you're looking at sensitivity to a stimulus, you're looking at some threshold, like how loud does a sound have to be for you to hear it, right? Because you can make a sound, so to speak, right? You can vibrate air, but perhaps the amplitude or the frequency is not appropriate for your auditory system to pick up on that, right? And so where do we have to get so that you can do that? Another thing we might do is what's the difference between two, right? Like how different do two colors need to be for you to recognize that they're two different colors, right? Obviously, if something's blue and something's red, you're like, okay, both of those are different. But as we start moving both of those, let's say, toward a purple, where does it become that you can't tell the difference in the two? There's sort of two ways, or a few ways we might do that. We might do something called the method of adjustment, right? So we'll give you one stimulus, here's purple, turn this dial until the other light becomes purple as well, and then we can kind of keep measuring how close you get it to that purple. That's one way to do it. Another way is something called a forced choice task. So what we'll do is give you 2 or 3 options. We're going to make you pick one. You pick the one that's different, right? The other couple are going to be identical, and in that way we can see how close can we get that oddball, right, to those other two before you sort of figure out what's going on. So here's sort of a example of that forced choice paradigm, right? So you have these vertical lines and then you have some oblique line, right? With the correct answer is picking the one that's oblique, right? The one that's not vertical. Pretty easy to do, right? I mean, most of you should be able to determine the correct answers. However, as we start to get correct answers, we slowly take something that was that orientation and we start making it closer and closer to that vertical orientation, right? Like how many degrees of difference do you need from vertical to know that this is no longer vertical, right? And how many times can you get that correct or incorrect? So we can keep asking you that over and over again. You think this video will play? It's an exciting video. Unfortunately, it's not going to play. This is, there's actually, if you can tell, it's a small animal right here. It's kind of difficult to see. Don't worry too much about it. The idea here is this animal was going to go pick out of these three circles which set of dots was moving faster, right? So two of these dots are going to kind of move kind of slow, and then one's going to move really fast. They'll keep picking that faster set of dots. Eventually, that faster set of dots will get slower and slower and slower and slower and slower until it can't tell the difference between any of those three, right? And then we know we found the threshold, right? Like, how much faster does a set of dots need to move in order for you to know it's moving faster than something else? Okay, you can create some kind of staircase, and then through some magical math, you can calculate a threshold. I'm not going to ask you to do this, but it's kind of important. Here's a guy, pretty exciting. Lots, actually, there's like a ton you could talk about when you talk about Alan Turing. In particular, probably the most important thing for us is his development of mathematical models, the way that he discussed computational devices, right, and thought about how you can think of the brain as a computational device, how we're breaking these things down mathematically, how can we create algorithms and artificial systems that can replicate what humans can do, right, which is kind of exciting. While there were some massive sort of advances made by him and some other individuals while he was doing his work, we're still not at the point where we've been able to create computers that can do the same things that humans can do, right? One of the big sort of areas of work here is in computer vision, right? Can we teach computers to look at a series of images and pull out images that contain certain objects, right? Some of you have probably seen there's like a Microsoft commercial where these guys like looking for snow leopards or something, right? And they're like, we got like 1 million pictures and it's going to take me 10 years to look through these. Stick them into my computer and magically it pulls out the 8 with a snow leopard in it, right? Or whatever, which is really fascinating. And while that is on some level extremely cool, on some other level that's very basic, right? Like, oh, there's a big leopard in this picture. Okay, I got it, right? When you're thinking about other things, you're looking at intelligence information, like satellite photos, right? Having an individual like human analyst look at those is very time consuming, right? If you had 10,000 images and you could feed that through a computer and you could pull out the ones that showed something interesting, that'd be cool. The other is medical imaging, right? So if you could, it takes time for a person to look through and find something, oh, that looks like a tumor, right? But if you can feed that through a computer and that computer is as accurate as a human, then you've really saved a lot of time. And of course, when you're dealing with things like tumors, time is life, literally. So that's where we're headed. We may get there at some point. There's this idea of a connectome. I'm not going to ask you too much about that. And then there's this guy, Jeff Hawkins. You guys heard of Jeff Hawkins? Most of you haven't. And I'm going to say something that like one other person in this room has heard of, probably based on their age. I'm just going to take an assumption the rest of you aren't quite there yet, Josh. How many of you remember the Palm Pilot? Anybody remember the Palm Pilot? Yeah, like a couple of you, right? Yeah, right. So this was, you remember the iPhone? Like that's a thing, right? So like before the iPhone, there was a thing called the Palm Pilot, right? Which was not a phone in any sense, but it was like a personal data assistant, right? In some sense. So it was kind of like a smartphone without the phone part. Then you could store a bunch of information in this thing, right? And Jeff Hawkins was the guy who did that. The cool thing about Jeff Hawkins is he actually runs a neuroscience institute now where they study different things about the brain. He did it the right way. He got into computers first and made a bunch of money, and then got into the science part, right? Like, so for those of you who are thinking like, hey, science is cool, brains are cool, don't do it the way like I did it. do it the way Jeff Hawkins did it, right? Because Jeff was like, hey, I'm going to make money in computers, and then I get to do whatever I want. I can just create my own institute, I can do my own research, and I can fund it. So I know, right, Aspen? Like he doesn't have to apply for grants. He is his own grant. I mean, he's his own money source, right? It's a fascinating thing. So I think, yeah, you should do it that way, right? So everybody change your major to computer science, and then once you've made millions and billions, and you're the next Bill Gates, then come back to us. We'll still be here. Things we can study with senses, we already figured that out. We talked about this stuff. You can have some more words there if you want to read them. There are some other concepts we should probably talk about. We talked about organization. We didn't talk about selectivity. You don't hear things through your eyes, okay? Your retina is not sensitive to sound waves. It's sensitive to light waves. It's very specific. And even deeper than that, there are specific cells that are sensitive to only certain wavelengths of light. And we'll talk about that selectivity as we go forward, right? Can you imagine, like, think about all the sensory information that's coming at you right now. Now, what if each one of your sensory receptors was sensitive to all of that information? What would you... Yeah, I mean, you wouldn't be getting anything, right? It's just like, that's all you would, that's your experience. It's just going to be constant, right? Like this constant, one single, like every tone you can imagine at one time, everything you could ever possibly see at one time, overlaid on top of everything else, right? I mean, I try to like take time to think about that experience and what that would be, and yeah, it's tough. Plasticity, things can change, right? Your brain's not infinitely plastic, but there are some things that can change from time to time. The other concept we really want to think about here is noise. And I don't mean noise in terms of like, your neighbors are having a party, right? That's noise, yeah. I'm talking about noise and signal, right? And how those two are related. So, how many of you buy your own produce? Yeah, so you're going to buy your produce, right? You want to buy some blueberries. And you pick up a pack of blueberries, right? And so there's some signal there. There's like good blueberries, right? But there's also some noise in there. There's some random like bad blueberries, right? Okay? And so you want to think about what's that signal to noise ratio, right? Like what's the important part coming through and what's just that random stuff that's happening, right? We talked about these action potentials, these electrical signals. Typically, they're only going to happen when there's some reason for them to happen, right? But on the other hand, they happen accidentally all the time, right? And so there's some like random firing of action potentials that just happens, some baseline, right? And you've got to get above that, right? So I think about this in this class, like some of you are going to complain. That's life, right? Some students complain. I don't know if you guys are aware of this. I'm not going to really change what I do unless your complaints get above baseline, right? Unless that signal gets above the noise. There's always some noise, right? 2 or 3 of you are going to complain about something. I don't really care, right? Because it's only 2 or 3 of you. That's baseline, right? If 12 of you complain, and so please don't, when they should try to get 11 other people together to complain about something, because it's not going to work, because I've told you the secret now. But if 12 of you complained about something, then I would think, well, maybe something needs, she's writing this down, like 12 people. So then you would think, well, maybe there's something really going on there, something that I actually need to look into, right? And so if you think about that from a nervous system perspective, there's going to be a random action potential here and there, but you're not going to get excited about it. All of a sudden you get a bunch of action potentials, you're like, that's a bear. I got to run from that thing, right? And so that's what's important. That's that signal to noise. The more noise there is, the harder it is to pick out the signal, right? Okay? And so we think sometimes about signal to noise ratios. All right, what else do we need to think about here? We've already talked about cortical receiving areas. That's not a big deal. Transduction, basically, this is very simple. We're taking a light wave, we're turning it into an electrical signal. We're taking a sound wave, we're turning it into an electrical signal. We're taking a molecule of salt, and we're turning it into an electrical signal, right? Whatever that sensory stimulus is, we've got to turn that into an electrical signal. Your brain runs on electrical signals. Here's primary sensory cortical areas, not too big of a deal. If you think about a bat, obviously got a bunch of auditory. Humans, not so much. Okay. Here are these concepts we already talked about. Not a big deal. We will talk about adaptation just a little bit in a couple weeks, right? How many of you have a dog or a cat? Yeah. How many of you don't recognize that? Like when you're home, but you've got people who come over and they're like, yep, that's rough, right? That's because you've adapted to that particular sensation, right? That particular experience. Someone else hasn't. How many of you have been away on vacation for a week or two and then come back to your cat? You're like, why did I ever keep this thing? It smells. Because you didn't know, right? But then by the time you unpack your luggage, you're like, oh, that cat's not so bad, right? Because you've adapted. So that's adaptation. We'll talk about why that's important later. Okay, so this is an important concept. I'm going to boil it down to something very simple. Adding one to a sensory stimulus does not always add one to your perception, okay? Right? Sometimes it adds more than one. Sometimes it adds less than one, right? And so if you think about that for a moment, sometimes as the sensory stimulus gets greater, you're going to perceive that it gets greater, but not every time are you going to perceive it as getting as much greater than you did the previous times, right? Does that make sense? So if I say tomorrow you got to write a 20-page paper, you're like, that's horrible. But then if I say no, it's going to be a 22-page paper, and you're like, okay, that's even a little more horrible, right? Because that's like a 2-page increase. But what if I said you don't have to write any papers at all, and then all of a sudden I say, no, a 2-page paper, right? So you went from like 0 to 2, which is like an infinite percent increase, even though it's only 2 pages. Or you went from 20 to 22, only a 10% increase, right? You don't feel it as much. That's Stevens's power law. It's not so bad, right? Don't worry about representation. That's going to get you confused. And then we'll get through the rest of this at some other time. All right. Questions, comments, concerns?